how the chromosome number is reduces. Do you want to know the answer? Let us start our chapter meiosis. So first of all, thank you for clicking this video. So today we are going to discussing about meiosis. So first term meiosis discovered by farmer Moore. So first term meiosis discovered by farmer Moore. Meiosis term discovered by Farmer and Moore. In animals, meiosis discovered by August Weismann. August Weismann. In animals. Now, what about plants? So, in plants, meiosis discovered by Strasburger. So, Strasburger. Meiosis occurs in all sexually reproducing organisms. During this sexually reproducing organisms, in this meiosis, first of all, meiosis means reduction division. What is a reduction division? So that's why during this reduction, fertilization can restore. So fertilization is restored. Means how it is fertilization is restored? If you see, during the meiotic cell division, get separated N haploid. So during the fertilization time, in the separated gametes are fused during the fertilization. We are getting again one organism. So like this, we are getting again one different organisms. So like that. This meiosis cell division also we called as heterotypic cell division. Why we called as meiosis cell division heterotypic cell division? If you see the heterotypic cell division means daughter cells are not identical to each other and parental cell. So that's what this meiosis cell division we called as heterotypic cell division. Now, the cell which is show meiosis cell division, the cell we called as meiocyte. Cell which is show the meiosis cell division we called as meiocyte. Based on the meiosis cell division, they are divided into three types. One is sporic meiosis cell division and gametic meiotic cell division, and another one zygotic meiotic cell division. So first we can see the zygotic meiosis. Zygotic meiosis. What is the zygotic meiosis? Here zygote is meiocyte. Here zygote was a meiocyte. So in which one is meiocyte here? Zygote. So for example, algae, fungi and thallophytic plants. What is the thallophytic plants? That is the undifferentiated plant body we call as thallophyte. So this is the zygote is the meiocyte. Zygote is the meiocyte. Sporic meiosis. Sporic meiosis. Spore mother cell. Spore mother cell was a meiocyte. Spore mother cell, here meiocyte. Spore mother cell is meiocyte. Spore mother cell. The best examples, higher plants. All higher plants comes under sporic meiosis. Spore mother cell. Examples, higher plants. Higher plants. One, that is the gametic meiosis. Gametic meiosis. Gametic meiosis. What is the gametic meiosis? So here gamete mother cell is a meiocyte. So gamete mother cell is the meiocyte. So gamete mother cell is the meiocyte. Gamete mother cell. Gamete mother cell. Cell is the meiocyte. Meiocyte. Examples, animals. Based on the meiocyte, meiosis, they are classified three types. First was zygotic meiosis, sporic meiosis and gametic meiosis. Zygotic meiosis. Here under zygote is meiocyte. Example, algae and fungi and thallophytic plants. Means undifferentiated plant body. Now sporic meiosis. In this sporic meiosis, meiocyte is spore mother cell. Example, higher plants. Now here is gamete mother cell. Example, animal meiosis. Right. Next we can discussing the silent features of meiosis. Can divide twice and chromosomes can divide once. So what is the main salient features of meiosis? Here nucleus can divide twice but chromosomes can divide one. So nucleus can divide twice and chromosome can divide once. Now when the meiosis cell division is start after parental chromosome, completion of meiosis cell division will start after parental chromosome replicated in S phase. So in this homologous chromosome we are observed and crossing over also involved in meiosis cell division. Homologous chromosome appears and crossing over was involved. After completion of this meiosis cell division, 4 cells are formed. In these 4 cells, parental chromosome, half of the chromosomes are parental cells and 1/4 DNA content. So now here pairing of homologous chromosomes, and pairing of homologous chromosome, crossing over was involved. End of meiosis, 4 cells will form. And in this cell, each cell, half of the chromosome was parental cell. 1/4 of DNA content. These are the salient features of meiosis. So in the silent features of meiosis, nucleus can divide twice, chromosomes can divide once. In this, pairing of homologous chromosomes will appear. Means, what is the homologous chromosome? If the chromosome is identical in structure and size and behavior, those type of chromosomes we called as homologous chromosomes. The chromosomes are identical in size and structure and behavior. Those type of chromosomes we called as pairing of homologous chromosome. Now crossing over will occurs or involved, means exchange of genetic non-sister chromatids. It is called as crossing over. So what is the sister and non-sister chromosomes? If the chromatids of the same chromosome, it is called as sister chromatids. Chromatids of different chromosomes we called as non-sister chromosome. So non-sister chromatids. At the end of telophase, at the end of meiosis, 4 cells are formed. In this, each cell is having half of the chromosome parental cell and 1/4 DNA content. These are the salient features of meiosis. Next we can discuss one by one stages of meiosis. So how many this meiosis, again they are divided into two stages. One is meiosis 1 and meiosis 2. And meiosis 1, again they are divided into karyokinesis 1 and cytokinesis 1. Under this karyokinesis, again they are divided 4 stages. One is prophase 1, metaphase 1, telophase 1 and anaphase 1. And prophase stage, again they are divided into 5 stages. That is leptotin, zygotin, packtin, diplotin, diakinesis. So after cytokinesis, meiosis 2, again they are divided into 2 stages, that is karyokinesis 2 and cytokinesis 2. Karyokinesis 2, again they are divided into 4 steps, that is the prophase 2, metaphase 2, anaphase 2 and telophase 2. So let us see meiosis 1, and meiosis 2. Meiosis 2. 1, again they are divided into karyokinesis 1, karyokinesis 1 and cytokinesis 1. So in this karyokinesis, again they are divided 4 stages, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. This prophase stage, again they are divided 5 sub stages, that is leptotin, zygotin, packtin, diplotin, diakinesis. How the prophase 1 is classified here? Based on chromosomal behavior, this prophase 1, again they are classified 5 phases. One is leptotin, zygotin, packtin, diplotin and diakinesis. So after completion of karyokinesis, cytokinesis 1 and next meiosis 2, again they are classified 2 stages. One is karyokinesis 2, again they are classified 4 stages, that is the prophase 2, metaphase 2 and anaphase 2 telophase 2, and cytokinesis 2. Two meiosis cell division. This meiosis 2 is similar to mitotic cell division. Now let us see one by one. Phase 1 stages based on chromosomal behavior to 5 subphases, that is leptotin, zygotin, packaging, diplotin and diakinesis. Now let us see leptotin stage. If you see the leptotin stage, the nucleus size will increase in leptotin stage. Nucleus size will increase. Slender and thread-like structure granules are formed. The granules we called as chromomere. So we called as the chromomere. Now if you see animal cell, in animal cell, chromosomes are arranged in scattered manner, means irregular. Chromosomes are arranged in irregular, means it is look like bouquet shape. So that's what this leptotin stage we called as bouquet stage. So while this leptotin we called as bouquet shape stage, if you see chromosomes are arranged irregular manner towards the centriole. That's what these chromosomes are look like bouquet shape structure. That's what this leptotin stage we called as bouquet stage. Now second one, zygotin. In this zygotin, the homologous chromosomes are visible. Means first of all, what is the homologous chromosomes? If the chromosomes is having identical in size, structure and behavior, it is called as homologous chromosomes. In this zygotin stage, homologous chromosomes get start together pairing. So homologous chromosomes get start the pairing. This process we called as synapsis. Synapsis appears in zygotin phase, and paired homologous chromosome we called as bivalent. So this paired homologous chromosome we called as bivalent. Homologous chromosomes comes together pairing, it is called as synapsis. So how the homologous chromosome will pair? Because of by help of synaptonymal complex. So how the homologous chromosomes will pair? By the help of synaptonymal complex. So that is these are the changes will appear in zygotin stage. Again here in this zygotin stage, chromosomes appears short, thicker in size, thicker due to condensation process. Due to condensation, chromosomes appears short and thicker due to condensation. If in animal cell, centriole duplication occurs in cytoplasm at S phase of interphase, and centriole duplication occurs in cytoplasm at S phase of interphase, can migrate at opposite poles and initiate the spindle formation if the cell is animal cell. Now next stage, packaging. So look on this diagram, packaging diagram. So bivalent appears tetrads. So bivalent appears tetrads. What is the meaning of bivalent here? Bivalent means the chromosome, two chromosome is having 4 chromatids. So bivalent chromosome means 2 chromosomes. This 2 chromosomes is having how many chromatids? 4 chromatids. Bivalent appears tetrad. Tetrad means 4. Tetrad means 4 chromatids. So that is the bivalent tetrads is clearly visible in packaging stage. Recombination nodules are formed. So what is the use of this recombination nodules? So when the crossing over occurs between the non-sister chromatids, so recombination nodules are formed by enzyme, digested recombinase enzyme. By the help of the enzyme digested by recombinase enzyme, in stage crossing over occurs. So what is the meaning of crossing over? If you see, exchange of genetic material in between the non-sister chromatids. It is called as crossing over. It is called as crossing over. This pacting stage is very longest stage. In this zygotin and pacting stage, both are very short stage. Pacting is a very long duration stage. Now, this is the chiasma. If you see, point where the non-sister chromatids are separated and fused. This particular part we called as chiasmata. This diploid stage is also very shorter duration stage. In this diploid stage, we are clearly observed chiasma is showing X-shaped structure. It is look like chiasma X shape. What is the chiasma? Point where the non-sister chromatids are separate and fused. It is called as chiasmata. Here chiasma, here clearly it is showing X shape. So this is the best identification for diploid stage. This is the best identification stage for diploid. And chromosomes, bivalent chromosomes separate to each other from each other due to activity, due to the repulsive activity. Next stage was diakinesis. In this diakinesis stage, what happens? A chiasmata can migrate towards the end of the chromatids. Towards the end of chromatids. This process we call as terminalization. What is the terminalization? Movement of chiasma towards the end of the chromatids. Towards the end of chromatids. It is called terminalization. In this diakinesis, nuclear membrane and nucleolus disappear, and bivalent chromosomes can migrate into the cytoplasm. In this, chromosomes are arranged scattered in cytoplasm due to disappearance of nuclear membrane and nucleolus. This is the diakinesis. Next one, metaphase 1. Metaphase 1. What happened? These chromosomes can migrate towards the equator plate. Towards the centromere of bivalent chromosomes. Centromere of this bivalent chromosomes can separate. This centromere of bivalent chromosomes present on opposite side of equator. Opposite side of equator. What happens? This metaphase 1 here. Chromosomes of bivalent chromosomes. Chromosome of bivalent chromosomes present on opposite side of equator. Opposite side of equator. These chromatids are located towards the poles. Chromatids located towards the pole, and centrioles of bivalent chromosomes present on opposite side of equator, and this chromatids are located towards this metaphase 1. Chromosomal congregation will appearance. Chromosomal congregation. What is the meaning of chromosomal congregation? In this meta of chromosomes from cytoplasm towards the equator plate. Towards the equator plate. That is called as chromosomal congressive. Chromosomal congressive. So what is the chromosomal congressive? If you see, the chromosomes can migrate towards the special character for metaphase 1. One bipolar spindle apparatus will form. One bipolar spindle apparatus will form. Now, one. So what happens? What type of characters we are observing in metaphase 1? Chromosome, bivalent chromosomes present on the opposite side of equator. Chromatids located towards the poles. Now, chromosomal congression means the chromosomes can migrate from cytoplasm towards the chromatids, equator plate, towards the equator. It is called as chromosomal congressive. Now next stage, that is the anaphase 1. So what happened this anaphase 1? If you see mitotic cell division anaphase 1, centriole separation will appear in mitotic cell division of anaphase. But here meiosis cell division of anaphase. So centriole duplication. So centriole division will not appearance in anaphase 1. So centriole does not divided. But what happened this anaphase 1? If you see, look on this picture. Bivalent chromosomes moves opposite poles. Bivalent chromosomes can move to opposite poles due to chromosomal segregation. So how the bivalent chromosomes are separated? Due to the chromosomal segregation. This bivalent chromosomes can move to the opposite poles. So here anaphase 1, another most important point. If you say diploid number of chromosome can reduces haploid number of chromosome. This is the best, this is very important in anaphase 1. So diploid chromosome number reduces haploid chromosome in anaphase 1. Next one, telophase 1. In this telophase 1, each cell is having nuclear membrane and nucleolus reappear. And each cell is having how many nuclei? 2 nuclei. Cell is having, after completion of telophase 1, if meiosis 1 may start cytokinesis or may not start cytokinesis. But meiosis 1 and meiosis 2, in between and interkinesis phase. What is the interkinesis phase? It is the very shortest duration phase. In this interkinesis phase, DNA replication does not occur. So DNA replication does not occur. After completion of telophase 1 it may start cytokinesis 1 or not followed by cytokinesis 1. But in between meiosis 1 and meiosis 2, interkinesis will present. So what happened this interkinesis 1 here? So interkinesis is a small duration. In this interkinesis, DNA replication does not occur. Next one, meiosis 2. So meiosis 2 is similar to mitotic cell division. Mitotic cell division. Look on this prophase 1. So already we studied mitotic cell division. So what happened this mitotic cell division? Prophase 2. In this meiosis 2, again they are divided into karyokinesis 2 and cytokinesis 2. Karyokinesis 2, again they are divided 4 subphases. Prophase 2, metaphase 2, anaphase 2 and telophase 2. First one, prophase 2. This prophase 2 is 2. What is the characteristic feature prophase 2 here? Chromatin material condensed. Chromatin material will condense to form a chromosome. In this chromosome can split longitudinally except centromere position to form a chromatids. Nuclear membrane and nucleolus disappear. Due to condensation of nuclear membrane and nucleolus, chromosomes can migrate from nucleoplasm to cytoplasm. These are the changes prophase 2. Next, metaphase 2. So what happened this metaphase 2? Metaphase 2, look on this picture. Two bipolar spindle fibers will appear. So this is the best identification for metaphase 2. Two bipolar spindle apparatus will form. What happened this and again metaphase 2? So chromosomes are arranging. Chromosomes can migrate into metaphase plate, and we are clearly observing two bipolar spindle apparatus. This is the metaphase 2. Now anaphase 2. What happened this chromatin, splitting of chromatin. So start division of chromatids in anaphase 2. Start division of centromere. So start division of centromere. Chromatids can move opposite poles. Chromatids can move opposite pole. But here anaphase 1 here, chromosomes can move opposite poles. But anaphase 2, Chromatids can move opposite poles. Chromatid can move. Next stage, telophase 2. So in this telophase stage, clearly observing cell is having 4 nuclei. 4 nuclei. At the end of telophase, chromosome condensation, decondensation will occur. Chromosomal condensation. At the end of telophase 2, 4 daughter nuclei will form. Next, at the end of telophase 2, cytokinesis 2 will start. What is the cytokinesis? Division of cytoplasm. Division of cytoplasm. Now, at the end of cytokinesis 2, 4 daughter cells are formed. 4 daughter cells will form. So, what is the karyokinesis and cytokinesis? Division of nucleus, it is called as karyokinesis. Division of cytoplasm, it is called as cytokinesis.